Catognatha. The word itself is from the Greek shot for long hair in gnathos for jaw, belonging to an exclusively marine phylum commonly known as arrowworms. Catognatha have around 100 species. These 100 species live in marine environments such as the tropics, polar places, and salty regions. 1/5 of the Catognatha population are benthic, meaning some live just above the ocean floor, but ultimately they do float and swim. However, reproduction in the Catognatha is quite unique. All Catognathas are hermaphroditic, meaning they have both male and female reproduction parts. They have large paired ovaries and testes, and the eggs are formed in the trunk region of the body, while the sperm develops in the tail portion. On the opposite end, the mouth is located ventrally on the head. The head bears on 2 rows of spine and are mostly holoplatonic predators, while they prey on their copepods. They are known for the serrated hooks or sharp teeth, enabling for success while catching prey. Their bodies range from 1 mm to 12 cm and are typically transparent, with 2 pairs of lateral fins and a caudal fin on the tail. Catognatha have bilateral symmetry and long bodies divided into head, trunk, and tail. The Catognatha are deuterosomes, referring to the anus appearing first, developing at or near the blastopore. The nervous system contains 6 ganglia that are located in the head, the cerebral ganglion, the 2 lateral ganglia on either side of the esophagus. Cold water Catognatha have a longer lifespan of approximately 2 years, rather than the 6-week lifespan of these creatures that live in warmer water. The Catognatha play an important role in the food chain, as they are mainly eaten by fish and are prey for larger carnivorous animals. As for the significance to humans, there really is not a relationship between the Catognatha and the Homo sapiens. However, they are used as indicators to detect the levels of water throughout the Atlantic Ocean in the North Sea. Next, as we move into the evolutionary history of the taxon, we can look at 2 different phylogenetic trees that we have built to help us come to some conclusions. The use of phylogenetic trees helps represent evolutionary relationships among species and can be generated by comparing ribosomal RNA. In the 16S tree generated using mesquite, the Catognatha, as you can see, is the 3rd down. Porifera and all other taxa on the tree have a common ancestor. However, Porifera is most related to the ancestor since it is the outgroup. In the literature, we found a tree that articulates the relationship between Catognatha and Echinodermata. The tree in the literature shows that there is not a relationship between the two, while the 16S tree shows that there is some relationship between the two. The literature also states that studies have shown that the Catognatha is a derived mollusk or nematode. However, the tree in our data matrix shows some relationship between the two, concluding that they come from a common ancestor. The closest relative in our 16S tree are the sister taxa, and the closest relative in our data matrix tree reads Annelida. The characteristics we chose for our data matrix might not have been the best because it is possible that they were too similar to the other characteristics listed. We chose 2 categories, the first being the eye. We picked eyes because they are connected to cephalization on specific lineages. Second, we picked the reproduction trait. We think that species that reproduce the same way should share a common ancestor, and those that reproduce differently delineate quite clearly. In conclusion, we generated 2 fairly different trees using our data matrix, which indicates that the data in our data matrix could have also had some errors.